o'clock, so I will make a start then. Right, let me press slideshow to go. Okay, perfect. Right, how are you getting on with the course? How are you finding the course material? Do you feel like you're getting through it okay, understanding it okay? Are you struggling a bit? How are you finding it? Good. Okay. You're finding it okay. That's good to know. Okay. I'm not sure that anyone else is typing. You can't always tell when people are typing on the chat. So I'll make a start, and if anyone says anything further, I can have a look in a moment or two. Okay. So at the end of last session, somebody asked a question, what was the difference between macrophages and fibroblasts? And I couldn't really answer it because they're very structurally similar. So they're functional differences, but I wasn't too sure what you'd be looking at in terms of structural differences because they're very similar. So I'm going to have a look at that in a moment. First of all, let me just say what we're going to have a look at today. We're going to have a look at this assessment criterion 3.3, which is distinguished between different types of tissue that are capable of regeneration and those that are not, we still need to cover today. So that's what we're going to be doing. But first of all, before I get into that, let me have a look at this and address the question that we were asked. So what was the difference between macrophages and the fibroblasts? So most of the ways you tell the difference between these two types of cells is just the fact that the macrophages are able to phagocytose stuff. So they, they undergo phagocytosis, they engulf things. So that's great if you're actually watching the cell and, take something in and phagocytose it. Great, you can tell what it's doing. You can see that it's a phagocytotic cell. You can go, oh, that looks like it might be a macrophage because it's able to carry out phagocytosis. But if it's not actually doing that, and you just have these two cells under the microscope, imagine they've been fixed. So when you look at cells under the microscope, they're often fixed so that you can look at them rather than moving, so they're not actively carrying out processes like phagocytosis. It's really hard to tell the differences. So let's have a look at some of the things that we might be able to use to tell the difference between macrophages and fibroblasts. Well, the shape. Okay, so this is an irregular shape, the macrophage, and that's basically because they need to be able to engulf pathogens and things like that. So they often have this quite irregular shape. They look like they've got arms or legs that engulf these pathogens. The fibroblasts are spindle shaped, so they're long-ish with tapered ends. So they should look different in shape, but you can't really tell what shape that macrophage is going to be in because they're so changeable. So the nucleus on both of these cell types are usually oval-ish in shape. So that's not really going to help you differentiate between these two cell types either, unfortunately. The cytoplasm, you might see some differences. So this can be a bit more granular in the macrophage. It depends how the cells are being fixed and stained if you're looking at them under the microscope, but you might be able to see some differences between the cells there. And the cell boundary, again, is a way you might be able to see some differences between these two cell types. So it says here that the fibroblasts are more well-defined compared to the macrophages, and that's because they're always like moving and engulfing things. But other than that, really, they're quite difficult to tell apart. These special features and mobility, you're only going to be able to really see, those differences if you're looking at cells which are in a state that are moving. So hopefully that answers the question. that was raised at the end of last session, but I realised there's not a huge amount to go on. It's quite a specialist job looking at stuff under a microscope, particularly under a light microscope. It's really difficult to tell because obviously the magnification is not that good on a light microscope. So it's quite a specialist job to tell the difference between these sort of things. But also, not really something that you need to be able to know or do at all for this course. So that brings me on to another point, really, is like the scope of this course, because this is an access course and it's roughly the same level as an A-level course, but an A-level course you would carry out over 2 years, and this one is a much reduced time frame. So the level of detail and depth that you will need to go in on this course is not really, really huge. And that will tie into your assessments, where you're asked to write about something and you're given relatively short word counts, 150 words, 200 words, for example. And it might not seem a lot given the depth and the detail which is in the written course material and the stuff that we go through in perhaps in the live sessions as well. But that's enough depth for this course. Believe me, when you go on to university, because you've gained access to your degree choice following this course, you go on to university, you will spend that first year with people that have got there from loads of different routes. So people that have done A levels, people that have done BTECs, people that have done V levels, maybe vocational courses in the future, and T levels as well, which are available now. So you'll be on your course with students that have taken many different routes, and that first year is spent making sure that everyone is essentially on the same page, because you'll all have done different courses and covered different content, and some people might have done biology A levels, and some people might have done chemistry and physics, and the course that you choose, they might go, do you know what, that's a science A level, that's enough, we'll let them on, but their biology might not be that strong. So the first year you spend all getting up to the same level. And if you've done a standard A-level course, you'll find you go over a lot of the content you did at A-level. And you guys similarly will find you're going back over a lot of the content that you did on this course in your first year of your degree. So don't worry that you need to know everything now. You don't. You will be introduced gradually when you get to university and go back over a lot of this stuff as well. So don't panic that you need to go into so much depth and cover all of these topics. And also be aware that we're only on the first unit, and a lot of the content that I've briefly brushed over, we will be addressing in more detail in other units as we meet them in the course as well. Okay, so we're going to have a look now at this last assessment criterion that we haven't covered just yet, and we are going to distinguish between tissue types that are capable of regeneration and those that are not. So why do we need to have tissue regeneration to start with? Well, we need tissue repair, and this means restoration of tissue structure and function after an injury. So we've all fallen over and cut ourselves as kids. We might have broken bones. It's that sort of stuff. It's repair. Now, it's possible to regenerate tissue, which is new growth, which completely restores portions of damaged tissue that has been lost to its completely normal state. So completely repaired, you can't see any damage, there's no scarring, or there's replacement. And this is where severely damaged or non-regenerable tissues are repaired by the laying down of connective tissue in a process that we refer to as scarring. So you might get a cut where you don't get a scar, because depending on the type of cut of your skin, or you might get a cut where it's taken a layer of skin off or gone deeper into the tissue where you do get scarring. So there's different types of tissue repair that we can get. There's either regeneration where it's back to normal, or there's replacement where you have some scarring. So if you get an injury, most tissue repair will consist of both this. So some of the tissue will be regenerated and be completely normal, and some of it will be replaced. So you will get some scarring, you get a bit of both. And what happens in the tissue repair is that some of the original structures of the damaged tissue, such as epithelial layers like the skin and the inside of your mouth maybe, will be repaired, but you may also get scar tissue formed, depending on what's happened. So myocardial infarction, like a heart attack, you might get some scar tissue. So the wound healing process depends very much on the type of tissue in which it occurs. So for this purpose, there's 3 different types of tissue that we could consider. We've got continuously dividing tissues. We call these labile, and these are cells that are constantly proliferating in order to replace dead or remove cells. So think about your skin, the epithelial tissue inside your mouth. You're constantly renewing the surface of these structures because they're getting worn away just from day-to-day living, eating, just protecting your body with regards to your skin. So these tissues contain pools of cells called stem cells, and these have enormous self-renewing ability, which gives rise to new cells of more than one different type of cell. So they can produce brand new cells. Then we have quiescent tissue, which are stable. So these are cells that normally exist in a not dividing state. So your skin and epithelial tissue is renewed a lot, but other tissues might not be dividing all the time. So they're just kind of, they're doing their thing, functioning as perhaps an organ like the liver or the kidneys, and they're totally fine unless they become damaged, in which case they can be induced to repair themselves. So an example, it says here, is the parenchymal cells of the liver, kidney, and pancreas. That means the actual functional cells of those organs, the ones that do the liver function stuff, the kidney function stuff. So, and then we've got the other cells, such as the fibroblasts and the smooth muscle cells. These are like the supporting cells of these tissues. So these would normally be stable and not constantly dividing, like, for example, the skin would be to be always replacing. But if you've got an injury, they could be sort of triggered to go into a repair type state. And then we've got non-dividing tissue. And these are composed of cells that have permanently left the cell cycle. I'll tell you what that is in a minute, and are therefore completely unable to proliferate. So these are permanent tissues like cardiac tissue or skeletal muscle tissue. And if these have to be repaired, there will be evidence of injury, such as a scar in these tissues. Oops, I've just got to move my little box from the screen. One sec. So we need to talk a little bit about stem cells, because when we repair tissues, these are where the repair comes from. And stem cells have a regenerative capacity. They're continually dividing. So they have the ability to self-renew, and they have the capacity to generate more than one cell type. So imagine this as a stem cell. It can divide, cell division, and produce 2 new cells. So that's really handy if you want to replace damaged tissue. Or you could get the situation where a cell divides. One of the cells produced could be identical to the parent cell, and one of the other cells, one of the daughter cells, we call these, one of the new cells, one of the daughter cells could have a slightly different function. So it could form a slightly different cell type. So stem cells have the ability to self-renew, to regenerate cells that are identical to the parent cell, or you can have something called asymmetric replication, such as we've got here, in which one of the daughter cells is able to self-renew. It's the same as the parent cell, and then the other one has differentiated. So it's specialized, and it can go off and mature into another type of cell, another cell type, essentially. So for these cells to heal after injury, they need to enter the cell cycle, essentially. So when you have cells in your body, they are usually in some part of the cell cycle, unless they're these cells that do not divide. So what's going on in the cell cycle? Well, through most of the cell cycle, the cells are doing their thing. They're growing, and if they're liver cells, they're doing detoxification things. If they're kidney cells, they're doing excretion things. You know, they're doing their thing. They're living their cell life, doing their function. So they grow during their normal life phase. Then they copy their DNA so that they can divide into two. They still carry on growing a little bit before the actual division phase. The division phase is called mitosis, the mitotic phase. And it's a really small part of the cell cycle, but you might remember it from school because it's talked about a lot because it's the phase where the cell is actually dividing. But for most of the cell's life, it is just growing, copies its DNA, ready to divide in two later on, and then another phase of just growing. So most of the time, the cell is just doing its thing, but there is this preparation for division here. Okay, so DNA replication and mitosis are parts of this cycle, and the continually proliferating cells, they're continually making new cells. Those cells are always moving through this cell cycle. The quiescent cells must be made or stimulated to go into this cell cycle. So external stimuli such as protein growth factors or cytokines, which are derived from the surrounding matrix. So these things would be stimuli or stimuluses if there was an injury. Okay, so these cytokines are part of the inflammation response. You don't really need to remember this particularly. I'm just telling you to make it make sense. So you get injured or you get an infection maybe. These cytokines, oh, you might have heard people talking about cytokine storms during COVID, depending on how much you were listening to the scientific reporting. Cytokine storms happen, part of the inflammation response if you get really poorly. So these cytokines will then trigger cells to enter this cell cycle. Things like the skin, epithelial tissue that needs to be constantly replaced, these cell cycles are just going on anyway. So the cell division bit, the actual mitotic phase at the top there, is a type of cell division where new cells are produced that are identical to the parent cells, which is great if you're looking to repair damaged tissue because you can replace like with like. So it's a continual process, but it can be divided into steps. We will look at this in more detail afterwards. This is just for information. You don't need to worry about it particularly here. You can see all the cells phases as it's going through mitosis here. So let me just have a look at the chat because I can see somebody's just asked me a question. Okay, Lana, no worries. All right, so these are the different phases of mitosis. This is all the same cell, just going through the division. And I'll just basically briefly go through what goes on. Step one, we call prophase. What's happening here is the chromosomes will become more condensed and compact, and you can actually see the chromosomes. You can't normally see them. And the cell membrane breaks down. That's what that little dotted line is there, the nuclear membrane. Sorry, I should say not the cell membrane. The nuclear membrane breaks down. So there's no nucleus at this point. And then we go into something called metaphase. And what you can see there is the DNA chromosomes. They line up in the middle of the cell. So metaphase, middle. And then we go into anaphase, anaphase apart. The chromosomes are pulled apart to the two ends of this cell. Remember I told you that the DNA copied itself, replicated before this division phase. So we've got copies. So one load of DNA there and the copies on the other side, for example. And then we get into telophase at the end where the cell reforms the nuclear membrane. We have two nuclei in our cell, and then it can divide by this process here, which is called cytokinesis. So you don't need to know all those names. We're going to talk about them later in the course, but just to show you that there's this continuous process of cell division, which can be induced to start to get these cells repairing themselves. So we're going to have a little look now at the regeneration versus scarring. And for tissue to go undergo regeneration without scarring, two things need to be happening. The injured cells must be capable of proliferation, so this making new cells, as is the case in this continually dividing tissues such as the skin or other epithelial tissue, or the quiescent tissues that can be stimulated to go into that cell division mitosis phase. And there must be an underlying framework intact. Yeah, so physiological regeneration occurs continually in dividing tissues, like we said, with the skin. The gastrointestinal epithelial cells are continually replaced by new cells from stem cells, like the mouth, I said. But pure regeneration is rare. It does occur with the liver. If you get acute injury from paracetamol overdose, it can repair. And if an injury only damages the functional, like parenchymal cells in a continuously dividing tissue, the repair and regeneration is possible. But if it's a very severe injury and it damages the underlying framework of tissue, then the regeneration isn't possible, and you get fibrous tissue deposited in the wound, so you get some like scarring. So now's a good time actually to talk about blood vessels and what happens in them and blood clots. So when blood tissues are damaged and the blood then can escape from the vessels, it will come into contact with new surfaces that it's not normally into contact with. And what happens then is it causes platelets to become activated and fibrinogen, which is soluble, to change. So platelets they're not actually cells, but they're found in the blood. So remember, we were talking about blood the other week. We said it's a connective tissue and it's mostly made up of this watery plasma. And when we were talking about connective tissue, we said that there are proteins in the extracellular matrix. You can't really see them in blood because they're dissolved. This is one of them here, fibrinogen. It's soluble, it's dissolved. And these platelets are not actually cells, they're bits of cells, they're bits of cell cytoplasm that are pinched off, but nonetheless they're floating in this plasma as well with this dissolved fibrinogen and lots of other stuff too, actually. So if the blood vessels are broken and the blood comes into contact with new surfaces that it's not normally in contact with, this activates some biochemicals that cause the platelets to become activated. And they also cause the fibrinogen, which is soluble, to come out of solution and to become something called fibrin, which is insoluble. So we've got these platelets, these little bits of cytoplasm that have been pinched off cells floating around in the blood now. And we've got all these fibrin fibers now. These are proteins which were soluble, but they're no longer soluble. These are also floating around in the blood. What happens next is these platelets, these little bits of pinched off cytoplasm, begin to stick together, they clump together, and they'll also stick to surfaces, so like the walls of the blood vessels and things. Then this fibrin, which is insoluble, so it's actual fibers now, will trap blood cells, like the red blood cells, and they'll form like a sort of mesh that will plug the wound. So platelets made with little bits of cells will then block off, essentially. They'll form a plug that the vessel itself will contract as well, because there's smooth muscle in the walls of the vessels which will contract. This is caused to contract, obviously, by chemicals. This isn't something we can control. It just happens as a result of the chemicals. And these platelets form this plug, which is reinforced by these fibrin fibers, and it will block any holes. They start to contract after a while, and this clot forms a tighter mass that pulls together and stops the blood escaping from the wounds. Stops you bleeding, essentially. So all of this is reinforced by more platelets and more fibrin. Let's have a little look at the diagram here. Oops. So here we have our blood vessel, which is severed, it's injured, and we've got all the components here leaving the vessel. So we've got the red blood cells, the erythrocytes here, we've got the white blood cells here, and they're leaking out. And you can see these tiny little bits of platelets. They're basically bits of cytoplasm as well. So the first thing that happens is the smooth muscle wall of the vessel can contract near to the point of the injury. So the lumen, the diameter of the lumen is much reduced. So there's much less space or volume for the blood to flow out. So that's going to reduce the blood loss. And also the diameter that's needed to be plugged by the fibrin and the platelets is smaller. So what happens next is the platelets are activated by chemicals that are released in the injury site. They become sticky, they stick to each other, and you can see them here forming a platelet plug, and they activate other platelets and cause them to move to the site and become sticky. So they move towards the source of the chemical signals, they bind together, and the plug will get bigger and bigger. And this fibrinogen, which is normally in solution, so it's normally dissolved, is converted into fibrin. It forms a mesh. You can see all these fibers here, and that produces a clot and stops any bleeding. Plug in the break there. You can see the actual fibers here, look. And these are the red blood cells in pink. So that's the fibrin fibers. So I thought I'd show you this actually because we talked about some of the stuff that was in the blood. This is the fiber here, the fibrinogen. These are proteins. But there's loads of other stuff. So we've got our white blood cells here, the B cells, the T cells, the phagocytes. Those are all white blood cells. We've got the platelets, which are not really cells, but they're bits of cells. We've got the red blood cells, which you can see so clearly over here. And then we've got this yellowy plasma liquid, which is mostly water. But there's an awful lot of stuff dissolved in it as well, including these proteins, the fibrinogen that we were talking about earlier, but the hormones we're going to talk about a lot when we get on to the homeostasis unit, the foods we'll talk about in the next unit, and waste products as well we'll talk about in the heart and lungs unit as well. So there's a lot of stuff in that blood. Okay, so a little bit more about the wound healing then. So the extent of scarring that we have depends very much on the type of the wound, and as we said, how deep it is, what sort of tissue is getting damaged. But let's have a look in terms of skin wounds, because we're all familiar with them because you get them all the time, you know, paper cuts, falling over when you're little, whatever. So let's have a look. So first intention healing is relatively small, and this is when you've got limited epithelial and connective tissue damage. So think like a paper cut. Yes, it hurts, but it's easy to heal. It's healing is rapid. The wound is small. Inflammation may be present, and scarring is absolutely minimal. And usually, if you get something like a paper cut, you can't see that there's been an injury there within a few weeks. It's normally pretty quick and not much damage. Then the other thing that can happen is second intention healing. This is where you get larger wounds. So the margins are not easily closed. So imagine a paper wound. It's really easy to stick that back together. I mean, they bleed a lot if you cut your finger, but you can easily stick the two ends of the wound back together. But if you've got a large wound where the skin area damaged is large, it's much more difficult. So example, if you've had a burn or an ulcer, or if you've had a graze, you know, when you used to go skidding across the playground and get a big graze on your knee, it was a large area, it would take a lot more time to heal and for the scab to form, because you can't just get the two bits of tissue back together. You've got this big open gaping wound, and you're going to get an abundance of fibrin and necrotic debris that basically is dead tissue. There's going to be a lot more inflammation. And so the scarring tissue or the granulation tissue is much larger. And if you think back to when you were old enough to be getting these grazed knees and in the playground, it would take a long time to heal. You'd have a scab, you'd have scars. You probably still got some scars from childhood. Most people do, you know, and that's because you've had a larger area of skin that needed to heal. Okay, so next little thing we need to talk about is stem cells, because we need to talk about stem cells and the role in embryonic development and possible therapies. So we're going to have a look at the different types of stem cells. So stem cells are also important, as we were just saying, in regeneration of tissue. So what we've got here is just very quickly after an egg and sperm have fused, what happens is that cell, so you get an egg and sperm, they fuse together, the nuclei fuse, the nucleuses fuse, and then you have one cell, and then that divides into two cells, and then they both divide and you have 4 cells, and then they both divide and you have 8 cells, they all divide, you have 16, and so on. So they divide by that process we were talking about earlier, that cell division called mitosis. So all of the cells are identical to the original cell that was formed when the egg and the sperm fused together. And don't worry too much about this either. We're going to talk about it again in another unit later. So at this point, each cell is genetically identical to the initial cell that was formed when the egg and the sperm fused. And that's what we have there. And then very quickly, we get to this stage. So here's your egg and sperm. They fuse together and they form a new cell. And then that will divide into identical copies. And then that will get to this stage. And then the cells of this ball of cells can then start to specialize. So the embryo will have a circulatory system, a nervous system, immune system, and so on. And if you have a look here, you'll see there's different words: totipotent, pluripotent, unipotent. And that basically, all these words are describing how many different cell types these cells can turn into. So it's not very easy to remember, to be honest, and I actually don't remember it off the top of my head very well at all. But they are important because they tell us specific things about the cells. So this specialization, which happens here at this blastocyst stage, is where the cells become what we call differentiated. They become specialized. So some of the cells are going to be part of the cardiac system, some are going to be part of the nervous system, some of them are going to be part of the immune system. It is really important for the development of the embryo, the fetus and the embryo. So how this happens is every cell in your body has all the exact same genes, but only a particular set of genes is switched on in particular cell types. And that's what makes them develop differently and have their different properties and then be specialized cells. We had a look at the specialized cells that were made up the specialist tissue, didn't we? So these cells that have the potential to differentiate into all these new and different cell types are called stem cells. So as you can imagine, these stem cells are really important because they've got the potential to become anything, any type of cell. And that's really important from a medical point of view, because if we've got cells that have got the potential to become heart cells or nerve cells or immune cells, maybe that means we can replace damaged heart cells or damaged nervous cells or damaged immune cells if we could somehow hack this process. So that's why we're interested in it as scientists, because of the medical potential. So let's have a little look. The 4 different cell types of stem cells. So adult stem cells are found in many parts of the body. And you might have heard when people have certain types of cancers like leukemia, they have like bone marrow transplants. So these are areas of tissue that are rich in stem cells. so they can replace some cells. But bone marrow cells can't become cardiac cells, for example, but they can become cells that are related to the blood and the immune system. So there's different levels of stem cells. Adult stem cells aren't as flexible as embryonic stem cells, because those embryonic stem cells we were looking at a slide or two ago, those ones that are found in the fetus and the embryo, can differentiate to become any tissue type. But adult stem cells are not as flexible. They can only differentiate into related cell types. So bone marrow cells can differentiate into immune system cells, so white blood cells essentially, but not other cell types. But they can be used to replace damaged or diseased or injured cells. And one of the main benefits of this is if you use stem cells that are taken from a patient, they can be reintroduced into that patient and not rejected by that patient's immune system. So if you were, for example, to give someone a blood transfusion of blood that wasn't their own, you have to be careful that they're not going to react to it. So when people I don't know, when they get an injury and they have to have blood in the hospital, one of the things that happens is they have to give them the right type of blood. You're probably aware of this, you know, type A, type B, type O. Depending on what blood type you are, you have to get a match. That's so you don't react to having blood that's not the same type as you. And similar things happen if you get tissue transplanted. So if you were to have a liver transplant or a heart transplant, because that's come from someone else's body, your body would recognize it as what we term non-self. That means not from you. They'd recognize it as being foreign tissue, and your body would start to reject it and produce an immune response against it. However, if you were able to grow a new heart from stem cells that had been taken from your bone marrow, for example, then you wouldn't reject it, because your body would recognize it as being part of your body, that your body would know those cells as being self, part of your body. So that's why stem cell therapy is so interesting and has so much potential, because we have the potential to get rid of that rejection of
I didn't get that.
Ooh.
Could you try again?
Sorry, just Siri kicking off again when I check the time. So it's going to get rid of that rejection, potential rejection of tissues, of bloods when we use our own stem cells. So that's why this technology has got so much potential. So here are the different cell types that we might be considering. So totipotent cells have the potential to differentiate into any of the cells needed to be able to grow an organism. So these are found in the embryo. And then we have pluripotent embryonic cells, which develop from these totipotent cells. And these are the precursors to the fundamental tissue layers of the embryo. So they have the potential to differentiate into any type of human tissue, but cannot support the full development of an organism. There's a lot of ethical issues with taking embryonic stem cells, as you can imagine, because these embryos are not really formed into an embryo. There's no beating heart or anything, but these cells have the potential to become a life. And there's a lot of argument as where does life begin? And you are removing the potential of a life. Where do we get these stem cells from? Well, usually from IVF. That's where we get them. It's because obviously when patients go for IVF treatment, they don't use all of the embryos that they make. So they can then be used for research. So there's a lot of issues there. Right, multipotent stem cells have the potential to differentiate into different types of cells within a given lineage or a small number of lineages. So when we were talking about cells taken from bone marrow before, we said they can be only differentiated into immune cells, for example. That's the sort of thing that we're talking about here. And then we've got oligopotent cells. These are multipotent cells that become further specialized and limited to one of only a few different cell types. And then we've got unipotent cells, which is fully specialized and can only reproduce to generate more of its own specific cell types. And these are often called parenchymal cells, so the actual functional cells, but we can reproduce them. So there's lots of different words there that are very, very similar, but each with their own unique meaning, essentially based upon how flexible they are in their ability to develop into different cell types. And why are we so interested in this? Well, I mean, yes, we could potentially cure baldness, which is kind of up there on the top right, which is a little bit superficial, but like blindness, deafness, heart attack problems, muscular dystrophy, diabetes, cancer, Crohn's, you know, these are some serious injuries and diseases that we could heal potentially with stem cells. So if we could get this to work well, it has got such huge potential for specialized medicine, which is tailored to the individual so that there's no rejection. And to just completely repair damaged tissues. And it could have a huge effect on aging as well. So adult stem cells are quite a good source of stem cells because they can be used to replace the damaged or diseased tissues in patients. And there's not the ethical issues of using embryonic stem cells. They have been used. There was effectively to develop whole organs. There was a trachea that's the windpipe that has been grown from stem cells and fully and successfully transplanted into a patient. Last time I looked into this, which was a while ago, they were working on a bladder, which again is a full organ that they were replacing. There's simple structures, you know, there's not a lot of, it's not a heart, it's not a lung, which are very complex structures, but there's still, you know, a large structure which would otherwise have to be transplanted. And because these cells were being taken from the patient's immune system with the trachea, what they were doing is they'd isolated some cells from the patient's body. They had a plastic scaffold, which was in a liquid that the cells would grow in, and they were sort of washing the cells over the plastic scaffold, and they were sticking together and formed a trachea. And then they removed the scaffold, transplanted the trachea into the patient, and it was successful. So it reduces the need for donor organs, which obviously is good because you're not having to take organs and pressure people into, right, I'm really sorry your loved one has just died the last 20 minutes ago, but can we harvest their organ type conversations, which are obviously pretty traumatic for loved ones because those always come from patients that have died a quick sudden death in an accident, like a car accident, you know. So it takes the need for that away, which is obviously good, and they're not rejected by the immune system. And obviously none of these issues with the embryonic stem cells. However, stem cell therapy hasn't always been very successful long term. So normally you see when you use stem cells or tissue that's been produced using stem cells, you normally see up to about 5 years, the results look really promising. But when you start to look at it longer term, it doesn't seem to be that successful. And we're not entirely sure why. And there are difficulty finding adult stem cell donors, for example, for things like bone marrow transplants. So it's not as easy as all that. And if you do grow cells in the lab, you know, so if you think, oh, I'm going to extract some of my own cells, grow them in the lab, after a number of generations, they do start to gain mutations. So that will affect the cell type. Obviously, that's not good. And it's quite hard to grow cells in the lab without them getting infected with viruses and fungi, actually, for that matter. It's such a technical procedure just to get in and out of these labs and stay sterile and clean. It's not as easy as you might think. Would religious views be considered part of the ethical disadvantages of stem cell use? Yeah, absolutely. I think that's where a lot of people get their morals and ethics from their religious background, you know. So yeah, I think this is a huge problem, especially in countries like, for example, I don't know, United States, where they haven't got such great rights for women. And they can be very anti-abortion and what they would, you know, pro-life, and they're like, when does the life begin? So some of these like quite fundamental religious societies like the US, there are quite a lot of issues with this type of technology. Yeah, definitely. Okay, so let's have a little look at some of these questions, and then we'll have a look at some of the ethics with this in a minute. So which statement is not correct out of the following? Which of the following is an ethical concern with stem cell therapy? And which statement applies to embryonic stem cells? So you don't have to put your answers up if you're not too sure and you're not feeling confident, but just have a little look and scribble the answer down for yourself if not. Okay, I will pop the answers up. So, which statement is not correct? So adult stem cells can differentiate into any cell type. That's not correct. They can usually differentiate into quite a limited stem cell type, which is very similar to the origin of that cell. Which of the following is an ethical concern with stem cell therapy? So embryos could be viewed as a commodity, not as an embryo with the potential to develop into a person. So people might start to see embryos as spare parts, and then women will be put at risk because eggs will need to be harvested. Vulnerable women would suffer. It's very difficult to legislate and to navigate morally and ethically. So which statement applies to embryonic stem cells? Well, they are all genetically identical when they're from the same person. So let's have a look at some of this ethics for embryonic stem cells. So they are sourced from embryos, which are produced for IVF and not used. So if you imagine a couple goes for IVF therapy, they'll produce several embryos and then maybe only one or two are implanted, and then you have a lot of embryos that aren't used. So they could then be destroyed, or they could be used for stem cell research. And each one of those embryos has the potential to develop into a person. So that's where the ethical difficulties begin. They aren't like this. You know, that is, I think maybe when you think of an embryo, that's perhaps what you think it looks like, but they aren't like that when the cells are used. They are literally a ball of cells. So it's not started to develop into an individual human. The cells are not specialized when they're taken. So they are literally just a ball of cells. But it's difficult to agree at what stage of development an embryo should be treated as a person. And some people say that life begins at conception, so when the nuclei of the egg and the sperm fuse. Some religious people say that. I think there's quite a lot of dispute as to whether that is actually said in the Bible, but a lot of the sort of fundamental Christians in places like the US believe that, so that causes a lot of ethical difficulties. A lot of the research in this area has been carried out in private clinics, and then they don't publish the results, and then the results aren't subject to peer review because they just treat it as this information as property of the business rather than scientific research that should be subject to peer review. They see it as business information that they don't want to share. So there are a lot of problems. However, therapeutic cloning could have huge advantages, as we've said, because of the very much patient-specific use of stem cells. What we could potentially do, in theory, is generate embryonic stem cells with the potential to become any type of cell in the body, and do this by reprogramming cells from our own bodies to send them back to that embryonic state. So in addition to these patient cells being used, they're from your own DNA. They'll be recognized as self rather than non-self. There's no danger of rejection from the immune system. So this therapeutic cloning has got a lot of potential. We could use stem cells in experiments to help us understand disease, and we could use them for treatments. And at this point, we've had no evidence that human embryos have been produced for therapeutic cloning, just for research purposes, and making sort of spare parts or anything. But I'm not too sure if it had happened, people would be publishing the results of it really either, because it's probably something they'd want to keep secrets too. So there's plenty of rogue states out there that could be carrying out research that wouldn't be approved of by the international community. So it's a very sketchy ground, really, some of this. So let's have a look. I think we're running out of time a little bit. So let's have a look at some ethical concerns with therapeutic cloning. So what I'd like you to do is have a little think about what you think some of the concerns could be here. What are some of the ethical concerns? Have a little think. Write it down. I'll give you a couple of minutes and then we'll go through. There's a movie actually which relates to a lot of this stuff called Gattaca. It's a really old movie now. It's probably like 30 years or something. But it's not actually cloning, but it's worth having a look at if you're interested in genetic modification and the potential where it could lead to. There is, I mean, obviously it's like sci-fi, but it gives you some sort of perspective. But have a little think, see what you can come up with that some of the ethical issues might be, and then we'll have a look in a minute. If you want to share some, that'd be good, but obviously you don't have to. Yeah, exactly. Does the donor give consent? Yeah. But even beyond that, you can imagine, what if you said, well, let's pay women to harvest their eggs? You know, how vulnerable would you have to be before you went, do you know what? I've got to feed my kids. I'm going to get some eggs harvested because I've got no choice. I'd imagine there's an awful lot of extremely poor women in a lot of countries that would see that as a viable option. So there's the exploitation thing as well as donor consent there, isn't there? Yeah, medical risks. It's not non-invasive harvesting eggs. It's pretty nasty. You have to have lots of injections to get your body to go through ovulation. It's not like getting a sperm sample from a man. It's pretty invasive medically. So yeah, there would be medical risks for sure. Let's have a little look at some of the issues then, because we are running out of time a little bit. So if we were going to do some therapeutic cloning, what we need to do is we need to create human embryos. So to obtain the stem cells, we need to create an embryo, and then we're going to destroy them essentially, because that embryo had the potential to become alive, but we're going to take away those cells to be stem cells rather than to become alive. So we've essentially created a life by fusing a nucleus of an egg and a sperm, and then we haven't let it become a life. So some people would see that as fine, it's just a ball of cells. Other people see it as murder, in the same way that they see abortion as murder. And I'm not saying abortion is murder, but like there's very strong views, particularly by some religious communities, for example. Look at the States, you know. So that leads us to the moral status of the embryo. Should that have the same moral legal status as a naturally conceived embryo? So think about the abortion argument in places like the States. When is it okay to abort a fetus? At what age does it become considered a human being? It's very, very difficult. Then there's the fear that some of these techniques could be used in therapeutic cloning and misused for creating cloned human beings, reproductive cloning. That's a pretty horrific thought. Think about cloning for armies and things like that. I know it's very sci-fi, but it has to be considered because there's the potential that it could happen, and it only takes some really rogue states for that sort of stuff to have potential. And then there's exploitation of women for egg donation. And I think this is probably the most concerning from my perspective, because I think it would be the most obvious thing that would happen, and it would then not be properly regulated, investigated. You could see this having a real impact on people. If a few embryos were destroyed, yes, it's an ethical consideration, and yes, maybe there's moral harm, but is there actual harm? There's a few cells that I don't think are at a point where there's even any pain felt. But this stage here, exploitation of women for egg donation, could have some very serious life impacts on people. And there are concerns about the unforeseen genetic or biological risks that could arise from manipulating cells early in the development. So there could be harm to the patients who have these cells used in their bodies. We're not sure. It's a very early stage in this sort of treatment. We don't know what the potential is for like cancers, maybe, for example. And then there's a lot of people that just think this whole process interferes with nature, and you're playing God by using this technology. Obviously not everyone believes in God, but it's a big deal for a lot of people who do, obviously. Okay, so we should have covered everything that we needed to cover for you guys to do these assessments now. Oh, that's assessment 2 that I've got there. The assessment criteria 2, but we should have covered everything. Oh yeah, because that's where the embryo one is. Define the pluripotent embryonic stem cells and their role in embryonic development, in the use in treating one health disorder. So use obviously the course materials. I know the word count is very small, 300 words. It's not a lot, but it's enough to get in the detail that you need at this level, because it's not, I mean, that's about a page of A4 handwritten, so it's not particularly short, you know, it's quite a bit of writing. But if you've got any questions, now's a good time. Or if not, I will see you on Thursday and we can pick up there. And if you've got any questions, then you can let me know there. Okay, thanks everyone.